All right, we're welcoming people in, and as we're welcoming in, we're going to go ahead and get started in about 20, 30 seconds. This is really just a terrific day. It's a beautiful day out here in Los Angeles. Again, my name's Dr. Timothy Fong. I'm one of the steering committee members, along with Chris Evans and Ziva Cooper, and we're bringing to you live on the Zoom our UCLA Cannabis Research Initiative Grand Rounds. We have about 4 of these per year. We'll welcome in a host of folks from UCLA Health System and beyond. Today, we're very, very excited to welcome in Dr. Daniele Piomelli. Dr. Cooper is going to give him a more formal talk, but he's going to be talking about paracannabinoids and chronic pain, and it has a whole bunch of information related to cannabinoids, which is exciting. We will have one more Cannabis Grand Rounds before the close of 2021. That will be in December. So watch for your calendars. Check our Twitter accounts, again, at UCLA Cannabis for updates for upcoming events. So with that, I'm going to turn it over to Dr. Cooper, who's going to introduce properly Dr. Piomelli, our esteemed guest from UCI today. So with that, Dr. Cooper, the Zoom is yours.
Great. Thank you so much, Dr. Fong. And it's really wonderful to have Dr. Piomelli here, truly an honor. And even if given the whole hour, I think it would be impossible to sum up Dr. Piomelli's contribution to science and impact on the cannabinoid field. So I'll try and sum everything up in about 30 seconds or a minute. Dr. Piomelli is the Distinguished Professor of Anatomy and Neurobiology, Pharmaceutical Sciences, Biological Chemistry, and Louise Turner Arnold Chair in the Neurosciences at UCI, right down the street from us. He's also the director of UCI's Multidisciplinary Center for the Study of Cannabis, and also directs the NIDA-supported P50 Center of Excellence, ICAL, the impact of cannabinoids across the lifespan. His training started with being mentored by 3 Nobel laureates, Eric Kandel, Paul Greengard, and Gerald Edelman. He was the first to elucidate pathways involving the formation and deactivation of brain endocannabinoids. So his work has been really instrumental in understanding the neurobiology and pharmacology of endocannabinoids. Dr. Piomelli has authored over 400 articles in top-tier, high-impact journals. He is the editor of the Cannabis and Cannabinoid Research publication that many of us receive. He's been the recipient of many national and international awards recognizing his scientific contributions. Other highlights on a more personal level is that Dr. Piomelli is an exceptional collaborator. You email him a question, he responds back with enthusiasm, curiosity, and excitement. Some interesting things to note about him. In his early days, he was a student of archaeology and linguistics. So he has a deep knowledge of antiquities, which is always surprising to me. And today, we'll be talking about paracannabinoids. You've heard about synthetic cannabinoids. You've heard about phytocannabinoids and endocannabinoids. Many of you, myself included, have not even heard about paracannabinoids until today or until you received the invitation to attend Grand Round. So I'm really excited to welcome Dr. Piomelli. He'll be talking about paracannabinoid control of the transition from acute to chronic pain. Thank you so much, and feel free to put questions and answers in the Q&A box.
Thank you so much, Dr. Cooper and Dr. Fong, for the too kind of an introduction. I'm going to share my screen now and get started. So as Dr. Cooper alluded to, I will talk to you today about paracannabinoids, and particularly about the role that this class of molecules has in the control of pain, as well as in the transition from acute to chronic pain. So as a starting point, let me just introduce this word. This is a term that is recently introduced in the literature. And by paracannabinoids, what we mean are naturally occurring lipid mediators that are similar in structure and in biogenesis, so in the synthetic and the degrading pathways to the endocannabinoids proper, anandamide and 2-arachinyl glycerol. But they have distinct effects, and sometimes these effects are ancillary or helpful to the effects of the endocannabinoids. And these molecules include palmethylethanolamide or PEA, olylethanolamide or EA, olylglycerol, and the N-acetyltaurines for now. Today, I will focus on the first one, palmethylethanolamide or PEA. And I'll start off going really back in time and to a clinical trial that actually a small set of clinical trials that were conducted during World War II, when proper medicines were, of course, in short supply, and doctors were trying to identify new ways of treating people, cheaper and simpler ways of treating people. They discovered that egg yolk alleviates inflammation. That observation led scientists to look into the basis, the pharmacological basis of this effect, and they discovered that it is the fat-like component, the lipids of egg yolk, that are responsible for these anti-inflammatory actions. And in 1957, chemists isolated one particular lipid, the compound palmethylethanolamide or PEA, and showed that this compound was responsible for the entirety of the anti-inflammatory actions of egg yolk. So here is the structure of PEA. For those of you who are into this kind of things, PEA is the amide of ethanolamine, which is here, and palmitic acid, which is this part here of the molecule. Palmitic acid is a fatty acid. It has 16 carbon atoms and no double bond. So it's a saturated fatty acid. So now this was, an interesting observation. Indeed, a few people picked up on it and showed that, both in animals and in humans, PEA has anti-inflammatory effects. In fact, there was a clinical trial back in the early 1970s that was conducted in a country that no longer exists, Czechoslovakia. Now is, of course, the two republics of Czech Republic and Slovak Republic, by a company that no longer exists called SPUFA. And what this company had done, basically, they synthesized PEA and tested as a prophylactic for acute respiratory tract infection. And this discovered in this paper, which is actually a really good paper, again, forgotten, unfortunately, but very, very good paper, that there was the compound PEA, which went by the commercial name of Impulsin, exerts anti-inflammatory effects in people. This was a fairly large placebo-controlled randomized trial. However, with the end of the Eastern Bloc, after the fall of the Berlin Wall, the Eastern Bloc rearranged itself. Czechoslovakia, as I just said, was split into two countries, and PEA disappeared. Meanwhile, back in the lab and in the United States, a famous pharmacologist by the name of Sid Nudienfriend, working with Nick Bachur, who was his postdoc at the time, discovered that actually PEA is a normal component of mammalian tissues. This is a table showing levels of PEA in various rat tissue. And that, of course, led to the conclusion that PEA is an endogenous compound with anti-inflammatory properties. The question that arose at that point is, how does PEA work? However, that question was not formally addressed. And in fact, it was only resolved by a chance discovery that my lab was involved in many, many years ago. And this was a work published in 1998, a collaboration with Professor Antonio Caldignano at the University of Naples. And I'm particularly glad today to bring up his face and his name, because today he got married to his 40-year companion and partner after, of course, a very long period of partnership together. So anyway, so Antonio and I showed something that is now been seen by many and replicated by many, that administration of the endogenous cannabinoid, endocannabinoid, and endamide, which you see here, the structure is shown here, by activating its own receptors called CB1 cannabinoid receptors present in the periphery of the body, so in the skin, causes a phenomenon of peripheral analgesia. So it is possible to, in other words, to block peripheral pain stimuli in the periphery of the body before they reach the spinal cord. And that is done also endogenously by anandamide. Now, in this particular paper, we were looking for control for anandamide. And again, you see anandamide here. I'm sorry if I insist on the structures, but that's kind of important. Anandamide is also, like PEA, is also the ethanolamide of a fatty acid. It's a different fatty acid. It's arachidonic acid, so 20 carbon atoms, 4 double bonds, but it's still a fatty acid. And here is the structure of PEA again. So the two compounds are similar in structure, different enough that PEA was known at the time not to activate CB1 cannabinoid receptors. So we used it as a control for anandamide in the hopes that anandamide would work and PEA would not work. Unfortunately, or fortunately, the discovery we made was that PEA does work. In fact, it produces a peripheral analgesia as powerful as anandamide does, but through a receptor that at the time was unknown. So that clearly brought PEA back into the limelight and raised the question, how does PEA work? So again, my lab at the time was working on another paracannabinoid. I mentioned it in my earlier slide, and it's a compound here, oleoyl ethanolamide. I don't want to belabor the point, but this is ethanolamine, and this is oleic acid, fatty acid, 18 carbons, 1 double bond, and showing that OEA is a powerful anorexic lipid mediator, controls feeding in animals and also in people. We know now for clinical, thanks to clinical trials. A few years after this observation, we discovered the receptor for OEA, which is this nuclear receptor called PPAR alpha. A few more words about that later on. But I wanted to point out to you that the structure of OEA, which you see here, and the structure of PEA, which I've now highlighted to you, are so similar that it was clear to us in the lab and to me and my students and postdocs that there must have been some connection. So I asked my then student, PhD student, Jess Laverme, who is now at AbbVie, to determine whether the anti-inflammatory effects of PEA, which have been known since the 50s, 1950s, were mediated by PPAR alpha. And in fact, part of his thesis work, it was this pharmacological research paper where he demonstrated that either by blocking the PPAR receptor with an antagonist or removing genetically the PPAR receptor, one could block the effects of PEA, and that PEA activates the PPAR receptor. So in addition to the anti-inflammatory effects of PEA, it turns out that PPAR alpha also mediates the analgesic effects. The two are connected, but they're not identical. And this paper here in JPET, which was a collaboration with several labs, including Andrea Homans and again, Antonio Calignano, showed that the analgesic effects of PEA proper are mediated by PPAR alpha. So a few words now about PPAR alpha so that we are all on the same page. So PPAR alpha is not a G protein coupled receptor. It doesn't look at all like, say, the opioid receptors or the dopamine receptors. It's a nuclear receptor, just like the receptor for retinoids or corticosteroids. So it usually sits in the cytosol of the cell, bound to a heat shock protein. When a ligand like PEA and OEA, but there are others, fatty acids, for example, they're not as high affinity, but they're lower affinity, but they're still ligands. When the ligand binds to the receptor, there are specific sites. The receptor sheds off the heat shock protein, which is kind of a negative regulator, brings in a retinoic X receptor, and as well as another co-activator called PGC1. There are two types, alpha and beta. And this complex travels to the nucleus, where it binds to responsive elements, and that causes reduced inflammation. On the other hand, this process also causes more rapidly, through mechanisms that are still unclear, reduced pain, possibly involving potassium channels, calcium-dependent potassium channels. So this is how PEA works, activating PPAR receptors. What is the interest of PEA in the regulation of pain? I showed you some really selection of very, very few preclinical data. There is a vast amount of data. It all goes in the same direction. But I think I wanted to raise your attention to the clinical interest of PEA. This is what I'm showing here is a meta-analysis, systematic review and meta-analysis published a few years ago on the efficacy of PEA for pain. And now the number of studies has increased, but there are about 2 or 3 more studies but basically about 1,000 patients right now. And the overall gestalt is that PA may be a useful treatment for pain, particularly for chronic pain, for chronic neuropathic pain, particularly for back pain. But I think that we still need some kind of pivotal trial. There have been placebo-controlled and randomized trials already, but certainly a few more trials would not hurt. In fact, we're conducting 2 right now at UCI, led by Ariana Nelson and by by Brent Young in the Department of Anesthesiology. So there is solid evidence that PA is a promising treatment for chronic pain, but it's also a fact that PA is not very effective, is not as effective as one would want. So in order for us to find ways to improving the effects of PA, we need to understand how it works. And here is a scheme to show, first of all, how PA may work in the regulation of acute pain, all right? So there are PPAR receptors in a couple of crucial cell types involved in the transmission of pain after tissue damage. And these are the C fiber, so these are small diameter sensory neurons and nociceptive neurons in the dorsal root ganglia, and these express PPAR alpha, but also macrophage of various types, both the endoneural macrophages in the dorsal root ganglia, but also resident macrophages in peripheral tissues also express PPAR alpha in fairly large amounts. And we believe that endogenous PEA acts by activating these receptors. Now, I just wanted to show you, just list one little piece of data that indeed there are PPAR alpha receptors in the DRG. This is a mouse DRG. This is a wild type mouse. And this is a mouse in which the receptor is being deleted by homologous recombination, just as a control for antibody selectivity. So there is PPAR alpha in the cell types that are involved in acute pain transmission, but does tissue damage regulate PA signaling? That would be important to show that indeed PA plays a role in the regulation of acute pain. And in order for us to answer the question, I'd like to just briefly overview how PA and this fellow OEA paracannabinoid OEA are produced. So this is a pathway that we and others have over the years characterized. There is basically, without going into too much technical detail, there is basically a family of lipid precursors that are sitting on the membrane typically, and these precursors upon demand, so when need arises, produce PA and OEA. So it happens that actually PA in particular is produced a fairly large amount tonically. So there is a fairly large amount of PA production that goes on tonically. And PA activates PPAR alpha, as we've seen before, just like OEA does. And then, like every signal that is produced, it also must be destroyed. And PA and OEA are destroyed by 2 main enzymes. One is called fatty acid hamide hydrolase, or FA, and that's an enzyme where my lab has spent an inordinate amount of time. We worked on them for 20 years, but I won't talk about it today. And the other one is an enzyme called N-acetylethanolamide acid amidase, or NA. And on that, I will come back in greater detail. So what these 2 enzymes do, they clip this amide bond here, forming palmitic acid, as well as, of course, ethanolamine. So the question that one needs, of course, to answer is whether tissue damage regulates PA signaling, because if PA is involved in the control of acute pain, then one would expect during painful situations to PA being somehow either produced or something else. And in fact, I'll summarize here many years of work, but what our lab and others have shown is that in healthy tissues, the levels of PA are relatively high. And by relatively, I mean way above the median concentration needed to activate PPAR alpha, which is about 3 macromolar. We typically have anywhere between 5 and 10 macromolar PA in the skin and in the spinal cord and in other parts of our body. So, however, during inflammation, something happens and the levels of PA decrease. So this is kind of a funny way of regulating a signal. most lipid signals, and you might be familiar to something called prostaglandins, for example, most lipid signals increase with stimulation. So you have tissue damage and they start being produced and then they decline. That's not the case for PA. PA is normally high and its levels go down. So it's really the other way around. And I just wanted to show you some data, again, just a nugget of data. This has been shown by many. In this particular experiment, we implanted a little sponge containing carrageenan under the skin of a mouse and measured levels of PEA in this inflammatory exudate that is produced. Carrageenan is a pro-inflammatory algae chemical. And normally, the cells that accumulate around the carrageenan sponge, which are mostly monocytes and polymorphonuclear leukocytes, they accumulate, they contain a fairly large amount of PEA. However, when carrageenan is added, so this is without carrageenan, when carrageenan is added, as you can see, the level of PEA drops dramatically and in a dose-dependent manner. So painful inflammatory stimuli lower PEA levels. And I just wanted to share with you the person who did this work, which was published in PNAS many years ago, Carlos Solorzano, who is now a venture capitalist, alas. He went to the dark side. So how does this work? How come PEA goes down after tissue damage? Well, we know, as I said before, that in healthy tissues, there is a fairly high amount of PEA, and this is due to the enzyme NAPE-PLD, which cleaves this precursor for PEA, NAPE, in fairly large amounts tonically. Then, PEA is destroyed by Na, but there isn't a whole lot of Na in tissue, so the product palmitic acid is pretty low. What happens in inflammation is that everything gets switched. So NAPE-PLD, the transcription of NAPE-PLD is suppressed. There is an epigenetic mechanism in the promoter of the NAPE-PLD gene that turns it off. And on the other hand, Na, the enzyme that cleaves PEA, is enhanced. Its expression is increased. So the net result is that you have less PEA and more palmitic acid. So you have less of the good guy, but palmitic acid turns out is a bad guy. I don't have time to go there today, but it's a pro-inflammatory and pro-analgesic compound. So basically, tissue damage weakens PEA-mediated signaling, which is analgesic, by decreasing PEA production and increasing PEA degradation. So I will focus on Na from now. For the rest of the talk, I have still a few minutes. And on the rest of the talk, I like to focus especially on the point as to whether Na can be used as a target for the discovery of new medicines. And the reason for that is because PEA works, but doesn't work very, very well. So that's why there are so many studies that are kind of borderline, I believe. The overall gestalt, as I said, is that it works, but one would like to see greater efficacy. And my own personal bias is that if we block the degradation of Na, which, as I told you, is increased, the expression of which is increased during tissue damage, we might be able to get more PEA and therefore a greater analgesic and anti-inflammatory effect. So we spent a significant amount of time on this particular enzyme, and which was originally cloned by Natsuo Ueda in Japan. And in collaboration with Bhushan Nagar at the University of Calgary, we determined its X-ray, the tridimensional structure by X-ray crystallography. And this is shown here without going into excessive detail. Basically, this shows, this part of the graph shows that there are two subunits in PEA, in Na. This is a signal peptide that gets clipped away pretty quickly. There are two subunits in Na, and there's a process of maturation of the enzyme, acid catalyzed maturation, whereby a cysteine in position 126 in humans, 131 in mice and rats, that gets cleaved, an amide bond gets cleaved, and the cysteine gets released. And that cysteine is the N-terminal nucleophile for Na. So it is a cysteine hydrolase, Na, and is a member of the N-terminal nucleophile family of enzymes. And this cysteine is shown here. So it is localized in endosomes and lysosomes because it requires a very acidic environment to work. Unless you put it at 4.5 or 5 pH, 4.5 or 5, it won't cleave almost anything. It is considerably expressed in immune cells as well as in neurons. And here, let me show you the expression in immune cells. This was work done by Alison Ribeiro and others a few years ago, showing that beta cells and monocytes express it in fairly high levels. T cells, granulocytes, and kappas and K cells don't express very high levels. And also shown here is that the transition from monocytes to macrophage, in this case, transition induced by forbolester, there is increase in expression. So this is a cysteine hydrolase. It is not that easy to make inhibitors for cysteine hydrolases, but they have the great advantage that unlike serine hydrolases, which are in large abundance, there are fewer. Cysteine hydrolases are fewer. So it is, in a way, it's although a little hard, once you get a good inhibitor, it tends to be more selective. And that was our hope when we started a campaign to discover inhibitors for the enzyme Na. And as of now, there are two main classes of such inhibitors. And those that bear the symbol ARN, the abbreviation ARN, are produced by my lab. And one class is covalent inhibitors, which interact with cysteine 131 or cysteine 126 in humans, and they interact covalently. So they form a thioester, essentially. You can see that these compounds are beta-lactones or beta-lactams, and they go by ARN 77 and 726. There are also non-covalent inhibitors, such as this compound, 1972, which we discovered in collaboration with a company down in San Diego, which is no longer interested in this kind of molecules. But I'll tell you a few more things about this molecule as we move forward with the presentation. I like just to mention that my good friend and collaborator, Marco Moore, at the University of Parma in Italy, is a leading figure in this work. So let me just say a few words about the covalent Na inhibitors. 77, you can see it here. This is a beta-lactone, substituted beta-lactone. The stereochemistry is really important to its activity. And 726 is a substituted beta-lactam. Also here, the stereochemistry is important. 77 is very potent. It's actually basically one digit nanomolar. It acts, as I said, by covalent Na inhibition. It's a very powerful analgesic and anti-inflammatory. However, it's only topically active because the beta-lactam group tends to open up due to the activity of peroxygenases in plasma. So one can put it on the skin, and it works very well as an anti-inflammatory and analgesic on the skin. In fact, it was moved to clinical testing for atopic dermatitis. Unfortunately, that trial was terminated prematurely. So 726 is a lactame, so it's a more stable molecule and not a great, not very much drug-like. In fact, we have stopped the development of this molecule, but we use it a lot to understand how Na works. And here is, in fact, the crystal structure. This is not a model. This is real. This is X-ray data. The crystal structure of 726 linked to this is cysteine 126 of the human Na. You can see here the formation of the thioester group, and this is localization of the chain of 726, which occupies normally the same space that is occupied by the acyl chain of palmitoyl ethanolamide. So this is where the palmitic acid group would be sitting. What about the covalent, non-covalent inhibitors? ARN 1972, the structure is shown here. It also binds very selectively to Na. You can see it here. This portion of the molecule, which is this part here. So this portion of the molecule occupies the fatty acid binding pocket of Na, whereas this sulfone group forms basically a hydrogen bonding with cysteine 126 or cysteine 131. And 726 occupies for the same cavity as PEA. 19702 is a very selective molecule. It's not extremely potent, but it's very selective. It's CNS active. Does not enter very much, only about 20%. The brain to plasma ratio is about 0.2, but enough to get to the CNS. It's reversible, and we proved that in a paper in Angewandte Chemie a few years ago. And as I will show you in a second, it's actually a really good analgesic. And here are the analgesic effects of 19702. For this experiment, we used the formalin model. For those of you who are not familiar with this model, I mentioned it before. Basically, it's injection of a small volume of formalin, which is an irritant, into the paw of a mouse. That causes a little tissue damage, and the mouse feels pain for a certain period of time, approximately 1 hour, 1 hour and a half. And so we measure basically the pain behavior. The pain behavior is a combination of licking, flinching, and a few other things. So basically you measure this nociceptive response. And what you see here is the nociceptive response. You will see that it's biphasic. In fact, that's a very peculiar thing about formalin. It causes an initial rapid phase within the first 5 minutes. And then there is a trough, and then it goes back up and stays up, but slowly going down for the next 45 to 60 minutes. And what is shown here is an experiment where we have administered 19702 by the oral route at the doses shown here, 0.1, 0.3, 1, and 3 milligram per kilogram, 1 hour before giving formalin, so to make sure that the enzyme was inhibited by the time we gave formalin. And what you see here is a very beautiful dose-dependent effect of 19702 on the nociceptive, or rather nocifensive, so pain-avoiding behavior of the animal injected with formalin. So okay, so 19702 is analgesic, which confirms the fact that PEA is involved in if you wish, in the signaling of acute pain as a protective mechanism. But I promised to you in my title that I would talk about something else, the transition from acute to chronic pain. And that's something that I think is extremely important, and extremely important to understand how relevant this phenomenon is. For those of you who are in the field, I'm really not breaking new ground. You know exactly what I'm talking about. Basically, a person undergoes surgery, say breast surgery, breast cancer surgery, or say thoracotomy, it's a heart surgery, open chest heart surgery, or even worse, it's a knee replacement surgery. And that person, feels very much in pain for the first couple of days, maybe the first couple of weeks. And then the pain in some people abates, and in others stays on for months and months, sometimes for years at end. And that pain is accompanied by everything we associate pain with: anxiety, stress, sleep deprivation, depression, loss of cognitive function. So we were interested in understanding if there could be a role for non-regulated PA signaling in the transition from acute to chronic pain. And I have to say, from a mechanistic perspective, very little is known about this phenomenon. In fact, so little is known, in part because there is no good model to study it. But I was particularly lucky that this young man, Yannick Fautior, joined my lab. Yannick is a native of Cameroon, but he he got his PharmD and PhD in Italy, and he joined my lab 2 years ago. He's done an amazing job. And with him, these other fellows, Alex Mabutani, who is also a Cameroonian, Kwang Muk Young, who comes from Korea, has been with me for 17 years. Francesca comes from Italy. Andrea Benaus is our token American. And actually, so is Andrea Homan. Andrea is a collaborator who has been working with me for many, many years. So the first thing we created a model for the transition from acute to chronic pain, a model that would allow us to actually have temporal resolution. So a model that will allow us to say, okay, the animal has this much pain and then develops this much pain later on. Okay, so quantitative and temporal resolution. What we did was very simple. We gave, again, the formalin, but instead of measuring the behavior for the first 90 minutes, we measured the behavior, then let the animals alone, and then measured again something else, which is called hypersensitivity, or in clinical terms, hyperalgesia or allodynia, to heat and pressure. So heat would be heat hyperalgesia, and pressure would be allodynia. So if we do that, and these are just heat hyperalgesia data, what we find is very interesting. So one injection of formalin, small amount, the animal no longer has spontaneous pain behavior that finished in the first 60 to 90 minutes, But now we have hypersensitivity. And we're measuring hypersensitivity on day 7, 14, and 60. These are 2 months later. In fact, we've gone all the way to 4 months later, and the situation is about the same. So this is something that lasts for a long time. In red, you can see that there is a very much decreased threshold to withdrawal latency, which indicates that these animals are in pain, but not for all animals. About 80% of them have it, and 20% don't. So we have 20% of animals that resist completely to this chronic pain stimulation. And please keep that in mind, because that's important. We do not know exactly how that happens. We have some ideas, but I want you to keep that in mind because it's important to follow the rest of the data. So if we now use imaging, and this is the diffusion tensor imaging, to look at what happens in the spinal cord. So diffusion tensor imaging, for those of you who are not familiar, measures fractional anisotropy, which is basically the tendency of water to go in a direction according to a tensor, or just not have such a direction. So there is an eigenvector. If the greater the eigenvector, the greater the anisotropy, essentially. So if you look at this and you look into the lumbar sacral region, what you see is that there is a decrease in anisotropy, which means that probably there is some damage, right? Focus on the red one for now. So there is some damage, right? And this damage is enough that water, instead of going in one direction, keeps staying around. Okay, I'm simplifying. But if you go now to the thoracic, and particularly to the cervical part, what you see is the opposite. So there is a change happening here, which likely means that there is a change in the structure of the spinal cord. There is something is happening in terms of either biochemically or either functionally or even possibly morphologically that is now detected by DTI and is a change in the opposite direction. So there is an increase in anisotropy. So these are the animals that I showed you before, are these guys here. By the way, these are about 100 animals here. You see 84. So, just to give you a sense of the fact that Yannick has no fear. He's a fearless guy. So these are the vulnerable guys. What about the resilient guys? The resilient guys, again, they respond to formalin. They have the quick response to formalin. That's normal. What they don't have is the persistent response to formalin, okay? And they have exactly the same lowering of the fractional anisotropy in the lumbar region, but they no longer express this increase in anisotropy in the thoracic and cervical region. Moving forward, if you go up, even more rostral to the brain, what you find is that the volume of different brain regions, and I highlighted a few here, the volume of these regions is specifically changed by invulnerable animals, so animals that received formalin in transition to this chronic hypersensitivity. For example, the infralimbic cortex, which is really important in regulating pain, but also in regulating the response to drugs, the volume is massively decreased. In the tenia tecta, which is involved in regulation of stress response, the volume is increased. There is in the right basolateral amygdala, there is a trend toward increase, and remarkably, in the CA2 region of the hippocampus, which is involved in social memory, there is an increase. And stuff happens also here in this fiber tracts. However, in the fiber tracts, I want to say right away that even our resilient animals have some changes in the fiber tracts. They don't have any such change in the rest of the forebrain, in the rest of the brain. So this appears to be linked to just the fact that the animal has been injected with formalin, whereas this appears to be linked to the fact that the animal has moved into this sort of transitional of this sort of chronic hypersensitivity. And this chronic hypersensitivity is actually a pain condition, and we can show this because they have anxiogenic responses in the elevated maze test. They have a cognitive impairment in the novel object recognition test, and they actually have loss in body weight gain, so that their weight trajectory is flatter, or it's actually completely flat. And these are all things that happen in people. Some people develop cognitive disorders, others more emotional, others have vegetative changes. Humans, of course, are much more variable, but we are capturing in this model what happens in the human chronic pain condition. So what about NA? So to study the effect of NA in the chronification process, we did not give NA as we had done before, the NA inhibitor, as we had done before formalin. But in the experiments I showed you before, we gave the 9 inhibitor before formalin. We saw that it inhibits the response to formalin, the acute response to formalin. That's not what we did here. What we did here was different. And this is, again, a testimony to Yannick's incredible fearlessness. He injected separate groups of animals on each one of these days, but only once. So the group of 10 animals received the 9 inhibitor, the 19702, on PFD1, another on PFD2, another group on PFD3, et cetera, all the way to PFD, post-formalin day 9. So basically, in every single day after injection of formalin, he injected to a separate group of animals and the NA inhibitor to see if there was any effect. And what he found is, I think, spectacular, because what he found is that if you inject the NA inhibitor on day 3 and 4, which is shown here, you are basically nullifying the effect of formalin. Normally, animals will remain in pain, as you can see here, for all the way for the first 9 days, and will. And I apologize. What I meant to say is that we measure this actually 2 weeks later. So the animals that have received the vehicle remain in pain 2 weeks later. The animals this is animals that did not receive formalin. These received formalin and vehicle, and these guys here received formalin and 19702. Those that received 19702 on PFD3 and 4 do not transition at all to chronic pain. Those that received it on PFD2, 2 days after formalin, transition only in part. That is, on about 50% of them transition. Those that received it on day 1, immediately after, the day 24 hours after, or on day 5, 6, 7, 8, or 9, after administration of formalin, they transition as if nothing had happened. And we're still giving them the 9 inhibitor, but they transition as if nothing had happened. Now, what does that mean? It means that there is a kind of a critical period where when we inhibit NA, somehow we are able to block the conversion of the creation, the establishment, the consolidation of this pain situation. But before we draw that conclusion, let me just show you some more data. So we look, this is DTI in the spinal cord, in the brain. Now, without giving you too much technical detail, basically we are now looking at whether the NA inhibitor is blocking the effect of formalin. So so the long-term effects of formalin. So we basically are looking for seeing this going down, which is exactly what is happening, and this going up, which is exactly what is happening. And in the brain, again, we're seeing things opposite to what we have seen before. And so basically we are seeing a reversal of the effect of formalin. So there is what we see in behavior, we also see in DTI morphological measures indicating that the microstructural alterations induced by formalin injection and the conversion to chronic pain are no longer there. And the same is true for the behavioral measures. I don't want to belabor the point, but basically NA inhibition between PFD2 and PFD4 blocks pain chronification. Now, what is important now is that not only NA inhibition does that. If NA inhibition does it, is presumably because PA levels have been normalized, yes. So what we did, we measured PA levels and we found that they've been normalized. So we gave it the PA or a synthetic PPAR alpha agonist, which is the compound GW7647 seen here. And we found that these two compounds, PA and GW, are able to mimic the effect of a NA inhibitor. However, and that is really what got me very excited about this, clinical use analgesics, opiates, NSAIDs, gabapentinoids, ketamine, even used at high doses that are known and do produce analgesia in our model, do not block the transition from acute to chronic pain. So this is where actually I had to push my postdoc a little bit more, because after seeing that, he was happy with the opiates and no, we have to test all of them. But he obliged me. And I think it's very, very important for a reason I will explain in a minute. So there is a critical period for pain chronification. And this critical period is between day 3 and 4. And at least in the mouse, at least in our model, right? Obviously in people will be different. And it will depend also on the stimulus, on the initial pain response. But at least in the mouse, under our conditions, what happens is that in the ipsilateral spinal cord on between L4 and L6. So we are cutting out that particular region that serves the paw that receives the formalin, right? We see an expression of Na. And this peaks at PND3, the day before the maximal, the first day of the maximal effect of the Na inhibitor. Presumably the protein is there at PND4, PFD4, and indeed we find the protein Na expression in spinal cord neurons ipsilateral to the damage. So what we did at this point is said, how the heck is this happening? How is it, what happens on PND3 and 4? Clearly a lot must be happening. And we were not disappointed because we ran a bunch of transcriptomic and metabolomic analysis. And what we found were really massive changes. It would take me 3 hours to go over them. But the key message is that there is a shift in spinal cord metabolism at the site of injury, ipsilateral, L4, L6. It's a real metabolic switch. And I don't expect you to look into this too carefully, but normally neurons, as you know, utilize as a source of ATP, they utilize oxidative phosphorylation, so mitochondrial respiration, the carboxylic acid or TCA or Krebs cycle for short. So this cycle here, they don't typically use a lot of glycolysis unless there is a need. And here is where the need appears. And what happens is that the switch is from the TCA cycle, mitochondrial respiration, to aerobic glycolysis. For those of you who are in the know of cancer biology, this is called this is a well-known effect that occurs in cancer cells when they need to do what? When they need to produce biomass to be able to multiply, to replicate. Because glycolysis produces pyruvate, which can be used as a source of amino acids, whereas the TCA cycle completely consumes pyruvate. So what is happening in the spinal cord is that there is a switch in metabolism at the site of injury from oxidative phosphorylation to glycolysis. And the function of this switch very likely is to generate biomass for synaptic plasticity, which we know very well occurs during chronic pain. This is what a lot of people have studied, changes in synaptic plasticity, something called the wound up, which is a form of long-term potentiation. But this effect here is causing the production of pyruvate, presumably, to allow that switch to happen, to allow synaptic plasticity to happen, but at a cost. The TCA cycle produces 30 molecules of ATP, 32 molecules of ATP. Glycolysis only produces 2. And so we have an energy crisis. In fact, we see accumulating IMP, AMP, which are signs of energy crisis, energy problem. So I am at almost at 45 minutes. I promised I would be within this time frame. So let me just come to some conclusions. So when in folks in which there is a transition from acute to chronic pain, and that again happens usually as a function of the intensity of the initial pain, the greater the intensity, the greater the likelihood that there will be such transition. It is the conclusion that we draw from these results that end organ injury causes within a certain time frame, which in our model is about 24 to 72 hours, may be different in other models, causes an induction in the expression of Na. And that induction in expression of Na causes a suppression in PEA signaling. But this effect is not now anymore linked to an analgesic effect as it was in the first part of my talk. Now, what this does is something different. It disables normal metabolism in the spinal cord by promoting a switch from oxidative phosphorylation to glycolysis through PPAR alpha. PPAR alpha is the receptor for PEA, and PPAR alpha is well known to be involved in the regulation of metabolism. In fact, that's the main function of PPAR alpha. So this metabolic switch, again, is needed. The spinal cord needs it because they need to support neural plasticity. They need to produce LTP, long-term potentiation, wind up. And for that, they need ion channels, they need receptors, they need plasma membranes. They need that. And we actually see in our metabolomic studies that they're producing those things. However, at the same time, they're producing less ATP. And as a result, this is an energy imbalance, and that we believe, and we don't have proof of that, that is at the basis of abnormal activity, ectopic activity that eventually leads to changes in the spinal cord. I remind you, the changes in DTI we observed, but also higher level changes in the frontal cortex, the prefrontal, particularly the ventromedial prefrontal cortex, maybe the hippocampus, maybe other regions that we have missed and others might be able to find. With that, I'll just say that, of course, if you block now, you're blocking this effect and you're blocking this switch. Now, we at this point must conclude, we don't have alternative to conclude that this is a novel pharmacological mechanism. It's not analgesic. They're not just analgesic. The inhibitors actually prevent brain chronification, such that if you were to give them, say after a high-risk surgery, the prediction would be that there should be no transition to pain. And we call this effect algostatic. This action, we call it algostatic. And the reviewer of our paper, which is impressive in Science Advances, had a big problem with this. But as I was telling Dr. Cooper before, I'm a stickler with words. And analgesic is one thing, and blocking, preventing the induction of pain or the consolidation of a chronic pain condition is a different thing. And the fact that classic analgesic drugs, the opiates, the gabapentinoids, ketamine, the NSAIDs, don't have the same pharmacological profile tells me that this is a novel pharmacological profile and which we have named algostatic. With that, I'll stop and thank you for your time. I'd be happy to take any questions. There are a few more minutes.
That was great. Thank you so much. Dr. Piomelli, before I forget, I also want to put in a plug for the UCI event that's happening on October 13th that will that you've organized to focus on the effects of cannabinoids and sex differences. So I'll send out a link for that. We have a couple of questions here, but I want to start off with one question. You were talking about how NOT inhibitors might be good after surgery to reduce pain chronification. And you mentioned that in the animal models, if you give NOT inhibitors prior to the formalin challenge, you can actually prevent the behavioral effects of pain hypersensitivity. I'm wondering, and maybe you talked about this and I missed it, but why not? Is there a reason why we wouldn't think about the utility of NOT inhibitors prior to surgery rather than immediately after surgery to potentially prevent the behavioral and morphological effects of pain hypersensitivity before the cascade even begins?
And this is a very, very important question. And I think it's a very important question because it goes to the core of something called preventive analgesia. Basically, preventive analgesia is something that's been explored, and if there are clinicians in the audience, I'm sure that they have heard the term. Basically, has been explored as a way of exactly preventing the induction of chronic pain. You are absolutely correct in that if you were to give a NOT inhibitor, presumably you will have, we don't know, of course, because no clinical trial has been done, but presumably you would have an analgesic effect property. It does, they do in animals very strongly. You saw the data. And there is a paper in JPEG just published by our group and others by many other groups. Interestingly, when we do our experiment after having given formalin, and we can measure pain because we can measure the pain response, right? We can measure the hypersensitivity response. It won't be spontaneous pain, but it's the hypersensitivity response. So when we give, say, ketamine or we give NSAIDs or whatever else, we see an immediate inhibition of the pain response, but then the pain goes back. The pain goes back. When we give the NOT inhibitor after, the pain does not go back. When we give PEA after, the pain does not go back. When we give GW7647, the pain does not go back. Ketamine, the other guys, they work, but the pain returns. So the key thing, in order to fully answer your response is to not just give the NOT inhibitor during surgery, but to extend the treatment after surgery sufficiently through a sufficiently long time to cover this whenever happens, this switch in humans. Because the key thing is that I'm convinced that if we do the same, we've done the same experiment in a different model of pain called the CCI model, in which one takes the sciatic nerve and gives a little ligation to that. And we also have the same sort of critical period, between 24 and 72 hours. But I'm sure that in people is different. I mean, we are different metabolism, different species. So I think the key thing would be, yes, to give the NAI inhibitor as a true painkiller, but also to give the NAI inhibitor afterwards long enough that it will prevent the establishment or consolidation of the chronic pain state.
That's great. Thank you so much. And so we have a question here from Juan Carlos Marvizan. He asks, this change in metabolism occurs only in DRG neurons or also in the dorsal horn? And how about how this might be affected by the spinal cord microglia?
Yeah, fantastic question. So first of all, I compressed a lot of data in a relatively short talk. In fact, if you see the paper, it will be out October 22 in Science Advances. If you see the paper, it has, I think, over 120 panels, data panels. Is just a phenomenal amount of work that Yannick and the others did. So I'm not going to fault you for misunderstanding what I said, because what I said was actually the metabolic switch occurs in the spinal cord. We did not look into the DRG, also because looking into the mouse DRG is really way, even for Yannick, would be too much. They're so tiny and they're so transparent and they're so difficult to find. You could do it in the rat. But in the mouse, what we know is that the ipsilateral L4, L6 dorsal spinal cord, so that includes both the dorsal and the ventral horn, there is clear increase in NAI expression, and there is clear changes in metabolic switch. The other question is the role, in what cells this switch occurs. Okay, well, thank you for asking that question, and hopefully I'll be able to answer it in 3 years or 4 years if our grant gets funded, because I'm waiting now the results of the resubmission of a grant on this very topic, and hopefully it will be in a fundable score. The point of the grant is to understand which cell types are involved in this phenomenon. And microglia, I believe, have an important role. However, there are certainly other cells, because the transcriptomic changes that we see have signatures, gene signatures that are clearly neuronal. The stuff that happens there is neuronal stuff. Not so much really immune. We don't see a lot of immune changes, some, but not as much as we see changes in metabolism and changes in ion channels, calcium channels, sodium, neuronal selective sodium channels, stuff like that. We also see a lot of stuff happening in the metabolomic panels. So microglia certainly play a role. Neurons certainly, I think, play a role. I think also we should keep our mind open. I think actually what happens is that there is a lot of penetration of peripheral cells into the spinal cord. In particular, what I'm thinking about is monocytes. Don't forget, when the microglia gets excited, they send CC2, and that drives in the monocytes. And in fact, in our immunohistochemical studies, we see these cells, which we have not identified, that kind of form a border, and they're all NA positive cells around the spinal cord. I think these are monocytes that are coming into the spinal cord to help the microglia deal with the cell damage. But anyway, and this is not an answer to your question, just a lot of blah, 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 but I will be able to answer it in a couple of years, because we have now started, and if it's funded, the grant is funded, we will be able to continue it. Single cell RNA-seq analysis, as well as cytoph analysis, to understand all this in minute detail.
Great. We don't actually have time for one more question, but I'm going to ask it anyway, and hopefully the attendees will be understanding. You talked a lot about PEA and NA inhibitors for chronic pain. We know that inflammation plays a role in a range of other pathological states. And I'm wondering if some of your research is looking at some of these other pathological states, mental illness that is associated with central nervous system inflammation, and the potential for some of these non-inhibitors to be helpful in those states, in addition to chronic pain.
This is, again, a very, very good question. I don't know. My thought, my suspicion is that yes, that they are. And the reason for that is because I believe that NA is really much more than just an enzyme involved in the regulation of PEA and pain. I think is really important at some basic level in cells like microglia, as has been alluded to before, and it gets activated when stuff goes wrong. And so I think that is quite likely that in conditions like depression, for example, where we know that there is extensive activation of the microglia in the brain, as well as, in fact, activation of monocytes in the periphery of the body. In those conditions, my suspicion is that NA will be found to be involved. We have not done any specific studies on that.
That's great. Thank you so much. This was really wonderful, and I've learned a lot, and it's made me think about a lot of future ideas maybe we can do in humans, looking at levels of PEA and OEA and NA in different pathological states, and thinking about how non-inhibitors might be good strategies for chronic pain. So thank you so much, Dr. Piomelli, and thank you very much for the attendees. And we look forward to seeing everybody in the future. And we hope to be able to post this recording at some point. Thank you.
Thank you for having me. Goodbye, everyone.